Hello everyone, this is Ashfaq Ahmed, and today we will cover about part 2 of how you can perform research in a protein or proteomics. So in part 1 we have covered most of the things, and just to give you a brief recap what we have covered in part 1, we have covered how proteins folds, how they perform functions, in fact what functions they perform and how they are performing. So I discussed these 5 methods in brief, and then I gave you some slight example of oligomerization, metalloprotein with some specific cases and conformational changes. And after that I talked about some protein interaction, and that was all about the part 1. Now in part 2 I will link all those things with the part 1, and then we will move on towards the practical tutorials. I know you people will wait for those practical tutorials, but these things are also essential. These introductory slides are also essential for you to understand very well. So let's start. In part 2 we will cover about thinking of a research project, or you can also say how and what to extract from a literature study. That means if you are doing a literature study, so what you can extract from it, or if you are given a task to construct your research project, so how can you construct your research project, what you have to focus on and how you have to focus on. So it's a continuation of part 1. If you haven't watched the part 1, I strongly recommend to watch the part 1, because I have covered most of the basic introduction in part 1. So this is a simple figure for biogenesis and abiogenesis experiment performed in Greek times. So a jar containing a piece of meat or flesh, and here the jar is open, here the jar is closed, here the jar is closed with some thin layer with pores. So and then they studied the formation of maggots, and at the end of the day, at that time they call it biogenesis or abiogenesis, and that means living things can be produced from non-living things or not. But in fact, if we see the same experiment in today's world, we may have such kinds of questions: what, who, where, when, how, why. So these are basically the 6 questions you need to ask, most of them during the literature study. So coming to the next slide, the first thing in a research is to construct your hypothesis and research question. How will you construct a research hypothesis and research question? I will first give you a daily life example, and then I will give you a specific example from the proteomic. If you see the left side, so everyone can observe something, some scenario. For example, you entered a classroom and that is too warm or too hot. So what you have observed, the classroom is too warm. Simple thing, okay? So a question in your mind will come automatically, that why is the classroom too warm? So, and normally then we also find out the answer, that why it's too hot could be because of the electricity, unavailability of electricity, could be because of the it's facing towards the sun, or it's construction fault, or anything you can say, but your mind will give you some answer. So you observe something in daily life, your mind asks a question, and then the hypothesis comes in. The classroom is too warm because no one turned on the air conditioning. It could be a possible reason that there is the air condition but no one has turned it on. So this is just a hypothesis. Okay, we call it a hypothesis to see if it is the real reason or not. So you have to make a prediction. If the air condition is turned on, then the classroom will no longer be too warm. This is a prediction because you have not tested yet. So whether it will make the classroom better or not, it will be tested. So you have observed something, then you have asked a question, you made a hypothesis in your mind, or you expressed that hypothesis, and then you did some prediction. So let's replicate this scenario in science, in proteomics particularly. Protein A is involved in cancer progression. For example, you have read somewhere, you have read on the internet or in some books that protein A is involved in cancer progressions. Okay, the question is, why protein A is involved in cancer progression? This question can also be asked this way: how protein A is involved in cancer progression? Okay, now the hypothesis is, if you are reading a literature, so you may come with such kind of hypothesis that protein A phosphorylate protein B. For example, there is a pathway where protein A is phosphorylating protein B, and that is leading, or that is helping the cancer progression. So this is our hypothesis to predict if protein B phosphorylation is blocked, cancer progression may stop. So for the prediction, you need to stop the phosphorylation of protein B, which is done by protein A. And if it affects the cancer progression, it means your question has a value, your hypothesis has a value. So by such manner, we generate our questions, we generate our proposals, we generate our hypothesis. But the question is, how such kind of observation in science will come in your mind? There is only one answer: by studying literature of that particular topic. If you do not study literature, so these kinds of observation will not come in your mind, or if it comes, it could be wrong. So you need to cover some literature study. Okay, now we are talking from the bioinformatics perspective. What are those techniques which can be used, or I would say which are most usable techniques nowadays? So the first one is structural modeling. What is structural modeling? For example, you are doing a research, you have a protein sequence, but you do not have a structure. So there is the need of structural modeling. Okay, normally people also call it homology modeling, but that is only one type of modeling. Okay, the second one is molecular docking. People also call it docking. Okay, the third one is simulation, or MD simulation. MD stands for molecular dynamic simulations. And the fourth one is machine learning. There could be many others methods or techniques, but I just grouped these four, which are, I would say, famous. And also if you are doing a bioinformatics research, so there are higher chances that you will interact with one of them or all of them. Okay, so let's talk about these techniques. Once you have a questions, once you have a hypothesis, so what are these techniques? So in this part I will give you just a brief slight introduction for my part 3, and in part 3 I will practically start the tutorials of these stuffs. Okay, for example, structural modeling. As I told you what structural modeling is doing. So structural modeling basically generates or predicts protein structure for you. Okay, so currently the Swiss model is working, the modeler is working, itaser is working, rubeta, tier rubeta, alphafold 2, and so many others, online and offline, or I would call it manual or automatic softwares are there, and you can consider it. So the first question is any method okay? For me, no. The answer is no, because some of the methods, for example, tier rosetta, the tier actually means transmembrane rosetta. So the tier is rosetta is basically designed for transmembrane proteins. So if your protein is a transmembrane and you are dealing with itaser or modeler or Swiss model, so you may hit a bad luck. Okay, so the same way, what is TM score? Many people are doing the or using the Swiss model or modeler or anything, but they ignore the TM score. So actually that is called template score, and it has a value in your modeling. So we will talk in about these things in next part, in the tutorial part, and what kind of structural mistakes you can have and how you can avoid those kind of structural mistakes. What are structural faults and what are incomplete structure, which should be oligomeric or with some cofactors, but you didn't model it. Okay, and so these 4 or 5 steps, actually these are different steps, or these are different precautionary measures you have to take care of when you are doing modeling. So in next video, in tutorial video of modeling, we will cover all these things in sufficient details for you. And finally, ProCheck is a program, most of the people are using it, and they will tell us that the structure is right or wrong. But ProCheck gives the information of structural quality. We call it stereochemistry quality, not the fold quality. Whether the fold is right or wrong, ProCheck is unable to tell you exactly. What I mean from this statement, for example, you are dealing with a transmembrane protein, and most of the transmembrane proteins, they contains alpha helical structures. So, and it will look like this. It should be look like this, a vertical alpha helical structure. So, for example, you are modeling something and it comes this like this. It shows you that like there are two helices and it will be in vertical manner and inside a membrane this way, but your structure comes like this. So if you see this alpha helix is just not a completely straight one. So in fact, ProCheck can give you a good result that this is a very good structure, but it may not be a biologically good structure, because the ProCheck gives you the data based on the stereochemistry, but you are dealing with transmembrane protein, and in transmembrane protein you may not or cannot have this kind of 0 degree alpha helix is lying on 0 degree. It somehow the preferred alpha helices are lying on the 90 degree. So the question is, my point I want to convey is that ProCheck can give you the stereochemistry quality but not the structural fold. It's something related to the literature study, I would say. And the secondly, most of the people are doing protein docking. We will also cover the protein docking, inshallah. So there are Autodock, Vina, MOE, and some online tool. But let me tell you, dockings are done for the two reasons. For interaction studies, if you have a question of interaction studies, so you can perform docking, and for protein inhibition. Sometimes you don't need to dock two proteins for interactions. you require to dock an organic molecule. Simply we can, not all organic molecules are drugs, but it's a fascinating word, so I will use a word drug. So docking your protein with a drug is for inhibition or slow down some process, is this part actually. So in inhibition we are targeting some proteins, and but you cannot perform this method blindly. First you have to identify that what is the region or the interface of interaction, what can we inhibit this protein. Remember, people are doing docking without studying the protein or without studying the proper literature about the protein, but there are certain proteins, we call it I would say housekeeping genes or housekeeping proteins. So if for example, if a protein is involved in the mismatch repair, but because of that some mutation in that proteins, disease, you are seeing a disease. Now if you are suggesting that by blocking this protein we will fix that disease, in fact you are not only trying to fixing the disease, but you are also trying to blocking that function which fix the mismatch of DNA. So in such cases, it is required that before docking you need to understand the nature of the protein. You cannot dock, you can dock, but it is not necessary that you fix each and everything with the drug, with the docking and with inhibition. You cannot inhibit each and every protein. There are too many other ways for treatment or for suggestion of treatment. So this is a very important point. This I just raised it here. So if you are doing a protein docking or protein drug or organic molecule docking, just just keep in mind that is it okay to dock this protein or not. And during the docking process we also perform some minimization before the docking. Why we are performing this minimization? Because most of the time the structure you are dealing with, you download it from the PDB, and the PDB structures are solved in the laboratory setup either by cryo-EM or crystallography or NMR. But remember, during crystallography or sample preparation, scientists are using different kinds of salts, like for example sodium nitrate, for example NaCl, for example calcium chloride, or so many other salts there are. So when they use salt, so it actually restrict protein fluctuations, and this way they can trap the protein into a single state. So the point is, the structure you are seeing is actually kind of restricted structure. So in minimization we are trying to remove all those, we call it crystal artifacts. So we trying to remove those artifacts, and therefore minimization is necessary for docking procedure, specifically if you are taking structure from PDB. Okay, the next another method I discussed there is MD, molecular dynamics study. So what are molecular dynamics? So there are a lot of programs I discuss it here: CHARM, Desmond in MD, OpenMM and GroomX. There are could be many others. So but all of them, they perform a uniform law or uniform stuff, which we call it application of Newton's second law. F is equal to ma. If you consider that mass is a constant, so force will be directly proportional to acceleration. And it's not only about consideration, actually it is a constant. For example, I'm hitting you slowly, you will have fewer lower acceleration, but I'm hitting you faster, or with a higher speed, so you will have a higher acceleration. Now in both cases your mass was constant for me, either hitting slowly or hitting faster. So therefore I told you that if you keep the mass constant, you will have force is equal to force is directly proportional to acceleration. So what we are doing in a simulation, we are making a system composed of ions, composed of water. The box, the kind of box is a water molecule, or the water molecules, and the greenish is actually a protein structure entrapped. And then we give some force to the system, and we allow the system to shake, we allow the system to move, and then the system produce some acceleration every atom in which is recorded. This is actually the basic philosophy of simulation, but we will cover it when we comes to the simulation part in tutorial. Okay, the next thing is interactions, mapping of interactions. This method can also help you in your research, and how you can map interaction, or I would say how you will find a different kind of interaction. So physical features there are, coevolutionary analysis there are. So there are programs, MATLAB based program like MALSIF. These kind of programs actually tells you that whether these two proteins are interacting or not based on the physical appearance or physical features. For example, if a protein has a totally hydrophobic surface and another protein has a totally polar surface, so maybe the MALSIF will tell you just for a common sense that no, they are not interacting. So on the other side, coevolutionary analysis. In the coevolutionary analysis, we actually required multiple sequence alignment and a bigger multiple sequence alignment. And there are programs like DCA, direct coupling analysis, PLM DCA, CMPRID, and based on the such kind of programs, you can actually predict the exact amino acid or the neighbors amino acid which will be involved in the interaction. And next on you can move on towards the docking to dock your stuffs with those amino acid. We call it interfaces. Okay, so what's next? If you ask me that what are the current research trends in bioinformatics, so I would say development of software for big data analysis. In my view, we have a big data of microarray data sets and RNA sequence data. Big data analysis through available language programs to develop a software for such kind of analysis is one current trend. To analyze such kind of data is another current trend. To use machine learning or deep learning algorithms, and for what? For drug designing. I will also cover these tutorials that how can you use machine learning algorithms for drug designing coupled with simulations and docking, and that will be very interesting for you. But wait, I will cover it later on. So designing and proposing a novel candidate drugs using these kind of stuff, and targeted simulations, I mean performing simulation. Okay, and tips for research publication. So I'm not covering this stuff right now. It will be some separate section, then we will cover it. And a final slide about a suggestion. If you are designing your research, so I would suggest do not design your research based on tools. Like before designing your research, if a student starts thinking that no, I will use docking, I will use simulation, now tell me which protein I should select. This is the wrong way. First, search your question, observation, hypothesis, then think about the tools, that what kind of tools I can use to address or to make some kind of prediction. That's why I told that avoid to generate questions by copying your colleague. For example, your colleague has done some docking studies and with a literature survey, and you are just changing the protein to just replicate that data. Maybe your protein is not suitable for docking, so it means you have just wasted your time. And at the end, someone or the reviewer will tell you that what nonsense you have merged, or what nonsense you have done. Why have you done docking on such proteins? And you will have no answer. To the reviewer, you cannot say that because my colleague has done this, so I just replicate the same thing. And the last thing, do not underestimate yourself or country facilities. You can if you will. It doesn't matter where you are sitting right now. It doesn't matter in which university you are. If you are determined, if you are focused, if you are energetic, you will and you can. Believe on yourself. So if you have any question, please ask your question in your comments, and your feedback are always important for me. If you like my videos, give it a high thumb, and subscribe this channel, and I can bring you more exciting stuff if you supports me. So that's for now, and bye bye. See you in next video. Thank you.